So our next speaker is Alyssa Bernstein. Alyssa is majoring in biology. She's a rising senior. She worked in my lab this summer, and she comes from Charlotte, North Carolina. And Alyssa, of course, is accompanied by Rosie. You might be surprised, or maybe not surprised to learn that when Alyssa was chosen to come this summer, Rosie is the first service dog that's ever been in the research labs at Mass General. So that was a lot of fun this spring to tell everybody and all the millions of different offices that Rosie is coming. But we really loved having both Alyssa and Rosie, and I think that we're all going to miss having a dog in the lab when Alyssa leaves. So Alyssa.
Hi, I'm Alyssa Bernstein. This is Rosie, and I worked in Sue's lab this summer. Today I'll be presenting on testing the in vivo efficacy of PTC 680, a promising treatment for familial dysautonomia. So familial dysautonomia is a sensory and autonomic neuropathy specific to the Ashkenazi Jewish population. Unfortunately, only about half of the patients survive to the age of 40. Significant to the mouse model that we used this summer, some significant symptoms include loss of proprioception, which leads to gait ataxia, neuronal abnormalities, and retinal degeneration. Here we can see that familial dysautonomia is caused by a splicing mutation in the ELP1 gene that leads to specific tissue-specific skipping of exon 20 and premature termination codon. The missplicing of this transcript introduces a frame shift in the transcript. Since every single patient with familial dysautonomia has this specific mutation, developing a splicing modulator therapy will be useful since it can be applied to every patient with familial dysautonomia. ELP1 is a scaffolding protein of the elongator complex, which is important to transcriptional elongation, translational efficiency, and cargo trafficking. In human cells in different tissues, genes splice differently with the most profound reduction in neuronal tissues. While the Solgenhop lab examines different aspects of familial dysautonomia altogether, my project was focused on the efficacy of this specific drug for treating this missplicing. Most recently in our lab, in order to study the pathology of the disease and test therapies targeting the splicing defect, the lab has generated a new phenotypic mouse model for familial dysautonomia by introducing the ELP1 gene, transgene with the familial dysautonomia splice mutation TGFD9 into an ELP1 hypomorphic mouse model, ELP1 delta 20 flox. This mouse model recapitulates the same tissue-specific missplicing observed in familial dysautonomia patients and the major phenotypes of the disease, which include reduced sensory deficits, gait ataxia and proprioceptive defects, and retinal degeneration. Here we can see an example of a genotype, and on the left we have the phenotypic mouse model, and on the right we have an example of a wild type allele. The tissue-specific missplicing of ELP1 appears differently in different tissues within the mouse model, including the retina, DRGs, brain, lung, liver, heart, and kidneys. Compared to the control mice, the FD model does have reduced growth rate, which is shown in this red line here, and the control is this blue line. Another symptom that familial dysautonomia patients have is spinal deformity. So here is the control, and here is an example of the mouse model, the phenotypic mouse model. In our control, you can see a normal retinal growth, whereas in familial dysautonomia patients and in the mouse model, they have reduced retinal degeneration or retinal cell growth. And in neuronal counts, there are reduced in DRGs, which I will go further into later in my presentation. One of my main tasks in the lab was using PCR and gel electrophoresis to determine human and mouse ELP1 alleles. For example, the first sample has this delta 20 and flox bands. So here's the delta 20 band and here's the flox band. And the second sample has the wild type band and the delta 20 band. And the third sample has the wild type band and the flox band. This first sample here is an example of an FD genotype mouse, whereas these are the control or phenotypically normal considering mice. The human FD ELP1 transgene is necessary in order for the FD mouse to survive. So before I came to the lab, there was analysis being done on a similar molecule to the one that I've been studying this summer, and that is PTC258. So they did gait analysis using catwalk, which I will also go further into a little bit later in my presentation, where they studied the stride length and base of support with control vehicle, FD vehicle, and different amounts of the molecule that was fed to the mice in their kibble. Here we can see that there were statistically significant results for the stride length of the front paws, the hind paws, and the base of support in their hind paws as well. Here we have images of the DRGs, or dorsal root ganglion cells. PV is parvalbumin, and it's a marker for proprioception, and that's what's shown, and it fluoresces in red, whereas NUN is an antibody that stands for all neuronal cell bodies, and that stains in green. And then here below, we have an image of the merged images. And then using that, we were able to identify the DRG volume and neurons per DRG. And as we can see here, there are statistically significant results that shows that PTC258 does rescue proprioceptive loss in the FD mice. So we expect the data from PTC680, which I've been studying this summer, to be similar to the data from PTC258 because it is an analog and so chemically similar to PTC258. One symptom of familial dysautonomia that we can visually see is using catwalk, which is this runway for the mice to walk across. So they go in this box and they walk across here, and it detects for gait analysis. So it also has a software to analyze the gait of familial dysautonomia mice in comparison to the control mice. And here we have the stride length and the base of support. So the stride length is the distance between the successive placements of the same paw. And here it shows an image of the right paw here, the right front paw. And then the base of support is the average width between either of the hind paws. And that's shown in this image here. So while we were working with preliminary data, we did find that the stride length for the left front paw was statistically significant, which we were expecting. However, because we're working with such a small sample size, and this is just preliminary data and there's still analysis being done, we did not find statistically significant results for the base of support. DRGs are dorsal root ganglion cells, and they're clusters of neurons that are found along the vertebral column and connect to the spinal cord. They're very important for sensory motor function, and the number and function of proprioceptive neurons and DRGs are reduced in people who have familial dysautonomia, as well as the mice model that we have. So in order to analyze this, we utilized immunohistochemistry staining and microscopy to visualize the DRGs. Here again is the parvalbumin staining, the NUN staining, and then here's the image that's merged. Below we have an image of the splicing of the PTC680. And as we can see that the percentage of the exon 20 inclusion increased from 15% to about 25%, which is significant. And we can see that in the graph model on the right as well. In conclusion, we have an excellent mouse model, TG-FD9-ELP1 delta 20 flox, in which to test drugs to treat familial dysautonomia. PTC258 and hopefully PTC680 will improve gait function in the FD mouse model. This summer, I was able to successfully distinguish different ELP1 alleles through PCR amplification, stain and use microscopy to study DRGs and utilize Catwalk software for gait analysis. I'd like to thank Sue and of course Colleen for keeping me healthy this summer as well. Elizabetta and especially Emily for working with me every day. And the rest of Sue's lab. Thank you so much for listening to my presentation. Are there any questions?
Practice with Rosie. She knew when your last slide came up.
She did. She was like, time to say hi to everyone.
We're done. I'm gonna go say hi to everybody. Any questions for Alyssa?
I was wondering how those two drugs differed in their ability to rescue the splicing.
So they're pretty similar chemically, but PTC 258 is less chemically potent than PTC 680. So while we did have good results from PTC 258, which was the original one, it can become toxic basically. So we needed something that was a little bit like chemically similar and would do the same thing, but would be more potent to the cells.